So good afternoon, everyone. As you will hear, I have an Australian accent, but I have it translated into English if you want to follow along there. I am going to be talking today about this thing called the BRD CAP, Bovine Respiratory Disease Coordinated Agricultural Project, which is a 5-year grant that's headed out of Texas A&M University by Jim Womack. And it really is a multi-institutional collaboration amongst a number of land-grant universities throughout the United States, and kind of broken down into research, education, and extension. And I'm the extension lead. Holly Nybergs at Washington State is the research lead. And really, what was the genesis of this project was trying to see whether we could use the tools of genomics to try to address selection for animals that are less susceptible to bovine respiratory disease, which is the biggest disease killer of both dairy and beef cattle in the United States. And so it's a disease that has an interest to both the cattle industries, and so this grant was focused not only on the beef industry but also on the cattle industry. And I think the afternoon session is really trying to target these traits that are not part of traditional genetic evaluations, because getting the phenotypes for them is expensive, or else the phenotypes are difficult to measure. Feed efficiency, reproduction, and I would argue disease resistance is also a very difficult one to get phenotypes on, and this is really not the ideal experiment to carry out for a selection program, and a very expensive project as well. So these traits typically can't be measured late in life or until the animal's dead, and are not routinely selected for because we don't have records on which to train the prediction equations. And thank you, Steve, for giving me a nice introduction to the training. And often they are traits that have low heritability. And so the reason that this project was attempted was the development of these single nucleotide polymorphism chips, or the SNP chips that we're all familiar with here, and the ability, theoretically, to dissect out host variation and possible genes associated with the variation as it relates to disease. But one of the things that's kind of prohibitive, or at least very costly as it relates to this, is we need to have very large populations to phenotype in order to be able to do a genome scan to meaningfully quantify genetic variation in these traits, especially when the traits are multigenic, that is hundreds or thousands of genes contributing to the trait of interest. And so it was understood that there would be a need to have populations that contribute or comprise thousands of animals. So this was beyond the UC Davis ability to do something with their 200 cow-calf herd or any other land-grant university on their own. And so what we've done is tried to partner with commercial operations that have the requisite number of animals in order to enable us to identify animals that were sick with bovine respiratory disease and have a partner or a pair that was not sick with the disease but had been exposed to the same environmental conditions. That was really what drove this. And when you're working with thousands of animals, you're typically using field data because other than perhaps US Mark, there's nowhere else that has this size of population that can be under one academic institution's control. And often this field data is very noisy, and part of the reason for that is that it's often left to a fairly subjective interpretation of the pen writer's determination as to which animals are sick or not, and some animals may be sick and appear healthy, and some may be not sick with BRD but get pulled in because they're looking depressed and don't have an appetite. And it's often unclear whether apparently healthy individuals have actually been exposed. And so they may actually be susceptible, but they just got lucky and didn't get in a pen where they were exposed. And so all of these contribute to making the genetic signal associated with the gene or with the trait less obvious. And so that basically decreases the heritability of the trait because all of these environmental factors are co-founding it. And so you have animals that would be apparently genetically susceptible that don't get sick, and that therefore kind of dilutes down the genetic signal. And that's one of the big problems with doing field data of disease information. So what was done was we found willing cooperators. They were willing when we asked them. I'm not sure if they would still be willing if you ask them now, but anyway, what they agreed to do in the case of the dairy is shown here, is to allow our team of, in this case, veterinarians from the veterinary centre down in Tulare to go and walk the pens of a Holstein calf rearing operation and look every morning before the treatment crews went out for sick animals. And we would tag those sick animals and then tag alongside them healthy animals. So animals that had nose contact with a sick animal, same environmental conditions, same breed, same herd usually, although the way these places work is that dairies bring in truckloads of their baby heifers and steers to get raised here. And what that basically gave us the ability to do is, although this may appear to be a chicken facility, these are actually each little pen here is a Holstein calf, and there's 70,000 of them in this one particular facility in Northern California. And so we could walk the pens and get really nice examples of animals that were very obviously sick with bovine respiratory disease, and its neighbor that wasn't sick with bovine respiratory disease. And one of the things that we tried to do with this project is develop an objective set of measures to identify whether an animal is sick or not, and take the subjectivity out of it. And to do that for the calves, we were able to use what's called the McGurk scoring system, which basically looks at 5 different attributes of a disease phenotype in these calves. Their temperature, which is a pretty classic one, whether they have a cough or not, and the way that this scoring system works is if you're totally healthy, you're going to get zeros the whole way down. You're going to have a normal temperature, you won't be coughing, you'll have no nasal discharge, no eyes weeping, and your ears will be alert and perky. If you're very sick, however, then you'll have everything the opposite. So you'll have a temperature, repeated spontaneous coughs, nasty nose, weepy eyes, and head on a droop. And I think this little guy is perhaps the most classic little BRD calf that you've ever seen in your life. And in order to qualify as a sick animal, you had to score at least a 5 or greater on this particular scoring system. And so that made it an objective approach to try to identify animals that were clearly sick. And then to be a control, you had to be a 4 or less in order to qualify. So a similar thing was done in collaboration with 2 large commercial feed yards for beef cattle. And I have to give a lot of credit to these feed yards, because when I tell you what they had to do for us, I think you'll appreciate that they were very much cooperators on this project. So we used pen riders to bring in animals that appeared to be sick, and then they would be rated on those 5 scoring systems, so whether they had ears, eyes, nose, temperature, and that would make it whether they were classified as a case or not. But then we asked the feedlot to bring in a healthy control pen mate that would be part of this experiment. And I'm sure you can imagine that that's not something they're excited to do. So we're bringing in healthy controls, getting blood off those animals, and also doing deep pharyngeal nasal swabs and collecting the bacteria that were associated with the disease. And then those animals were from there on housed together and killed together. And again, that was a big ask and something that I really appreciate the 2 feedlots doing. There was one feedlot here in the Midwest, and then there was another one out in Washington that were collaborators with us. And basically between the dairy cars, we had 2,000 cases, 2,000 controls, and the feedlots, we had 2,000 cases and 2,000 controls. But we screened thousands of animals to get that subset put together. And I'll just give you a summary of where the research is. So the researchers are still combing through the data. We've done all the phenotype collection and the genotype collection on these animals, and have done 800K SNP chips on all the animals, and then done the GWAS, the Genome-Wide Association study, looking to see if there are markers that are disproportionately associated with animals that are either sick or healthy, depending on whether you're a glass half full or half empty kind of person, that would be indicators of susceptibility to bovine respiratory disease. And I'm just going to put the preliminary results for the BEEF study is that they found that the heritability estimates that they were finding in this binary case control phenotype was around about 17% and 30% when using a numerical value of the McGurk system as a semi-quantitative definition rather than a yes-no kind of case control definition. And so those estimates of heritability are pretty high for disease traits. Typically disease traits are below 0.1 or 10% for heritability. And so the research team believes that's because they were able to objectively identify these animals and have a less subjective approach to identification of animals that were sick or not. And they're currently trying to narrow in on what are apparently regions of the genome that seem to be associated with susceptibility of BRD, and actually sequencing those pieces to see whether or not they're able to identify causative QTN or quantitative trait nucleotide, that is the SNP that's responsible for an animal being less susceptible to bovine respiratory disease due to the fact that they have this particular allelic variant present in their genome. So that is still ongoing. This project's 5.5 years into a 5-year grant, and you might say, well, that sounds like it should be done, and it should be, but you get this 1-year extension, and we're running a little bit behind on the research, and so at this stage I can't give you a nice little finished cookbook, we're done, this is what we found, because the research team is still working on trying to identify these QTNs. And so for me, I guess as the extension person, it's hard to know what to do if you don't have the finished results to then extend. And so a lot of this is a little bit hand-waving because the approach that's taken to integrate this information will very much depend on the genetic architecture of this particular trait and indeed feed efficiency and even the reproduction. So if there are many loci, as in thousands, that have little tiny effects, the infinitesimal model, which tends to be what we find in animals with traits that are we're interested in, economic traits like growth and the like, then we're probably, as we do with those other traits, going to have a genomic prediction approach. And as such, we'll need to have ongoing phenotyping such that we can keep updating our prediction equations, similar to what was discussed by Steve this morning. The other hope is, and this is where I think it's fair to say that the jury's out as to what the result of this is going to be, is if there is the possibility of identifying these single nucleotide QTNs that have an effect on susceptibility to disease, the hope is that if they're actual functional nucleotides, that is, whatever it is about that particular base pair makes the animal less susceptible, that is going to be a functional variant that's going to be seen across breeds. So we're no longer talking about genomic prediction equations, we're really talking about actual functional variants like DGAT that was discussed this morning. Now DGAT's a beautiful example, but it is a very, very, very big effect gene, and there, as was mentioned earlier, there's not that many very big effect genes that have been found up to this point, and so whether we'll find it for BRD or not, I'm not sure. If the number of QTLs that we have do boil down to QTNs and we're able to explain even 20% of the variation in susceptibility, it's not everything, but it's a lot better than the nothing we have now as our selection criteria for BRD, because basically we're not including this in our selection programs at the moment because we have no selection criteria. And so at the moment I'm not sure which of these two will be the correct model, and which is the correct model will affect how we'll incorporate it into our genomic evaluations, and also whether we need to have ongoing phenotyping for breed-specific prediction equations if it's the form of the polygenic effect. And I think we really don't know at this stage what the outcome is going to be. Perhaps for dairy it's a little simpler. Everything's a little simpler in dairy, partly because one breed is such a dominant force there, and because they already have an ongoing program to kind of update their prediction equations all the time. We potentially could incorporate it into their prediction equations as they exist right now. If there are SNP markers that are associated with big effect, obviously they could go on the chip and they could be incorporated into evaluations. But what is perhaps the thing that is most important and that's currently not in there, and it relates back to Matt's presentation this morning, is how to appropriately incorporate this trait into their selection decisions. And in the case of dairy, they have a single cumulative index that's used to select their animals, the dollar net merit index, which basically looks at all of the different economically relevant traits for which we have selection criteria and puts an appropriate economic weighting on those traits. And for those of you that don't do too much with dairy, I put up here the evolution of the USDA economic index for dairy cattle, starting in 1971, going through to the most recent index in 2014. And what you see here is an evolution of the development of genetic approaches to improvement of dairy cattle and an ever-increasing number of traits that are known to be economically important, and their slow integration into the index that is associated with dairy. So back in the olden days, it was just milk and fat. That was it. That was the only thing it's not exactly single trait selection, but it's pretty close. And then as the technology became more sophisticated and there was appreciation of the value of protein that got included, then we started to have some things like productive life. So okay, you make a lot of milk, but if you're only there just lactation, then you're not as useful as someone that's there for a longer time. And you can see the addition here of somatic cell score, which is associated with the disease trait of mastitis, the udder composition, the feet and legs, the body size, and then fertility starts to come in here, down here, and finally calving ease, till we get over here to an index that's currently being used. And what I think is really interesting is, and I use this quite a lot in public lectures, is that the dairy index currently has a -1 rating for milk production. And so this is a balanced index that looks at what actually are the economic drivers of dairy production, and obviously fat and protein are positively correlated with milk production, so that kind of comes along with selection there. But I want to really draw your attention to this somatic cell score. So somatic cell score is proportional to disease incidence of mastitis, so you want lower somatic cell score, which would decrease your incidence of mastitis, so hence the negative weighting here. But the incorporation of it first started in 1994 in the index, and then it's had about a 10% weighting thereafter. And I want to have a look at what the genetic trend of this particular disease trait is, using the indicator trait of somatic cell score in an index where it's not the only thing getting selected for. It has a slight negative emphasis on this particular trait. So we're still selecting for productive, healthy, fertile cows, but we don't want the ones that get mastitis. And I think this genetic trend score is pretty enlightening. This is a heritability of 0.08, the average somatic cell score. And here's where they started putting some selection pressure against it at 6, the weighting of -6. And then you can see the rating of -9 got incorporated there. And you can see, as a result of that, this very steep decline in the somatic cell score, and there's a resultant decrease in mastitis. And so you can make improvements in disease traits, even while still making selection to improve the other traits of economic importance, by including a selection criteria that has something to do with that trait in your index. And so I think that's really good news and a really nice positive story. Of course, it is a little easier in dairy, where there's a milk recording group that goes out and collects that as part of the Dairy Herd Improvement Association. And so you basically get that as almost an auxiliary data point from doing the milk analysis and the milk production levels. And so they're quite fortunate that they get that as part of their typical testing. So how can we do something like that in beef cattle as it relates to bovine respiratory disease? And I wanted to talk about this project that I've been working with Simplot, where we've been having a look at the potential of using genomic information in a vertically integrated commercial beef cattle enterprise. And this is basically a partnership between Simplot Davis, Mike McNeil, Delta G, and GeneSeek, to try to see in an integrated beef operation how might genomics get incorporated into that type of an enterprise. And basically our objectives were to try to develop genomically enhanced EBVs using data collected on ranch for commercial calves in the feedlot for the selection of their terminal sires. To determine the cost benefit, that's always my interest. Does it make sense for commercial producers to invest in this technology for an operation where they're actually retaining ownership and actually getting the value from improved genetics back to the same group that's making investments in genetic improvement, and then have a look at other opportunities for deriving additional value from genomic information, such as potentially marker-assisted management. And I bring this up as it relates to BRD. So basically what we've done is we've been collecting genotypes and phenotypes from the calves on this operation for 3 years now, and we've been using the integrated chips where we basically get the DNA at the same time that we put an EID into these animals and a unique identifier. So none of the calves anything happens to them before they come to the feedlot. That's the first time we see them. And when they come in, they get this put onto them. And the calves we're working with, I guess maybe Simplot has smoky coloured barns because we're just working with these Charolais sired terminal offspring, so the smokies that are shown in this picture here. So basically anything that's got that colour, it comes onto our project. And it's all gets integrated. It's quite a sophisticated system, and some of you that have done some genotyping this will look familiar to you with the envelopes and the different pieces of information, trying to get all the data together on each calf. But basically we run these calves through the chute, we get a measurement on them, we take DNA off their ear tags, we tag them, there's a crew that works to do all of that, and really this was initiated by my master's student Brian Welly. It was going to be his PhD project, but then the vet school got him, and so now it's a postdoc's project, and we basically now have information on all of these calves, and this is the phenotypes that we've collected on about almost 13,000 animals. So gender, polled status, typical kind of feedlot traits, but the one I want to draw your attention to here is BRD. And so we had almost 13,000 animals go through, and we had about 1,189 treated, so what's that, about 10% treatment rate for BRD. And we recorded whether they got BRD and also how many treatments they got for BRD. And we used all of the data that we got from that facility to get real estimates of what the cost of each case of BRD was to that facility, not only in terms of treatments and veterinary costs, but also how it affected their intake, how it affected their hot carcass weight, how it affected their overall grading, both for marbling and yield. And we did all of that and worked it into an economic model to give us some sort of a feel for what's the appropriate economic weighting to put on selection against BRD. So we know what it's worth to have an extra pound of weaning weight. What's it worth to have a decreased percentage of BRD incidence? And the results are just recently accepted by the Journal of Animal Science. And basically what this table shows is the relative economic value of increasing or decreasing these different traits by 1 unit. And so the way that you could read this is for every decrease in incidence in BRD on this operation, there's a $2.08 return to every animal that's on that entire operation. Are you right, Dare? And so if we look at the relative weighting of this, it's actually, here's the lowest relative economic value, days to harvest, it's actually the second most important relative economic value in that feedlot index, which is a terminal index for this particular operation. And each % increase in morbidity is associated with an average loss of $2.08 per head across every animal that's in the feedlot, right? And so it's actually a very important economic trait. And according to this, the weighting to select for animals that are less susceptible should be quite high in any type of a terminal index. This is obviously one location with one set of cost factors, but it does give us a very real look at what on those 13,000 animals, what the relative economic weighting of this particular trait was. And so the fact that there is variation in resistance to disease, coupled, I would argue, with increasing consumer pressure to address disease in a way other than the use of antibiotics, makes it an incredibly attractive solution to try to incorporate selection against disease in our indexes. And even if the markers don't predict all of it, if they predict even 20%, I think given the economic weightings that we found in that commercial simulation, that we would actually want to incorporate that into an index and have a selection criterion where at the current time we have none. And so when we look at incorporation into beef evaluations, it's a little bit different to dairy. And so again, if there's large causative effect mutations, that theoretically will be translatable across breeds, although obviously we'll have to determine whether that's actually the case. Otherwise, we're going to need to develop prediction equations in the same way we have for marbling and all of the other traits of interest to beef production on a breed-by-breed basis. And that's going to mean that each breed is going to have some phenotypes of bovine respiratory disease upon which to basically do their calibrations and their ongoing refresh of their genomic prediction equations. This is not the nicest outcome, but it may be what the data tells us. So I think this would be ideal, it'd be nice, but I don't know what the outcome's going to be. So in order for this to happen, we need some type of an objective approach to BRD disease diagnosis that can be used by breed associations, maybe with their sire test progeny or with a group or subset of animals, where they can have an objective determination as to whether an animal got BRD or not, and make it a little bit less subjective than the current approach. And so a group was put together to try to develop some standardized guidelines for BIF that would help to enable the production of EPDs for bovine respiratory disease on a breed-by-breed basis, and to add to the accuracy of selection against that trait, and also to allow validation of newly developed genomic panels. And that's one of the problems we have, and I'm sorry I used the word validation, I guess I'll use the word, what was it? I forget what that other word was, but calibration or evaluation of newly developed genomic panels to see whether there's an association between those and actual disease incidence in the field. Because at the current time, there's really very few groups that have the data for testing, although I might argue there's a fairly large group here that has data for testing that type of thing when they come to the evaluation stage. So as I mentioned, the current diagnosis tends to be this DART approach, so depression, appetite, respiratory rate, and temperature elevation, and that's really very dependent on the competence and diligence and astuteness of the pen riders. And this is the symptomatic animal at the moment is typically got an observation of eye discharge, depression, lethargy, and a rectal temperature of greater than 103. And actually, part of this project, we had indwelling rectal thermometers in some of our cattle in Davis. And for pretty much the whole month of July, the rectal temperature never got below 103 because it's just hot out. And so this arbitrary cutoff is really not very sensible when you look at environmental conditions, especially in hot weather. And all of that makes these systems somewhat subjective in nature. And so there's a number of different ways that we can record BRD. We can say yes, no, got it, didn't, binary, based on DART, which is dependent on skill level of the rider, an objective scoring system like the McGurk, some type of combination of systems, or more sophisticated measurements such as the Whisper stethoscope system that you may be familiar with, where you can listen to the lungs to determine whether it sounds like they're congested. But ultimately there has to be some type of a balance between the amount of data that gets recorded and the benefit that comes back from it. So if we put forward an impossibly complicated list of symptoms that have to be collected on any animal that's part of this evaluation program, we're not going to get very good compliance because it's going to be too hard to do. And so what the group did was we said, well, what's currently getting recorded? Because we know that there has to be a balance between the effort required to collect the health data and the subsequent benefits, be they genetic evaluations, which would be ideally what we'd like to come out of this. And of course, Electronic systems make such data capture easy. That's what we used on that commercial example that I discussed earlier. But there's obvious gains that we could get in reliability for low heritability traits, such as diseases, but we need to make sure that there's the appropriate balance here. And so the BRD Guidelines Committee for BIF got put together, and Larry, who's in the room here, is on there. Some veterinarians from here, and also Jim Lowe, who's a veterinarian from the University of Illinois, who's actually also involved with the group that developed the whisper technology. Then some of the researchers on the BRD CAP, and myself and Mark Enns are the group that had a look at this. And what we said was, well, what's getting recorded now? What are the feedlots recording? And so we looked at data from two widely used feedlot software programs, and the production animal consultants provided a summary of what gets reported. And so basically, this is the information that gets reported on all animals that go into these feedlots using these particular recording programs. And so certain things you get 100% recording, not surprisingly, whether in, out date and weight, owner, goes down a little bit, origin, starting average weight 100%, starting head, ending head, breed, zero. For the most part, they are not recording breed at the feedlots. And that's really a problem if we're trying to do breed-specific evaluations of BRD. And so clearly, if we're going to do this, we're going to need some type of mechanism to keep track, especially of breed association animals going through feedlots, in order to get that data back into breed evaluations. And then this is the data that's typically recorded on treatment information. And so the date of the treatment is recorded, weight for the most part, temperature, 75% of the animals get their temperature recorded. Which products they get treated with is 100% compliance. That makes sense, doesn't it? That you want to know what you treated so you can have the appropriate withdrawal time on there. And typically you get a diagnosis, but sometimes that diagnosis is I don't know, or other. And so that diagnosis is really based on the person evaluating that animal at the shoot. And so it may be a very skillful person that's very accurate in their diagnosis, or it may be Joe Blow that really doesn't know too much, and if they've got a runny nose, they get treated with an antibiotic and it's BRD, just because otherwise they might go back to the pen and get sick, and then they'll get in trouble because they didn't notice an animal that was sick. And so this is a somewhat subjective information, but that's basically what's out there now. So ideally what we would like is a recording of the temperature, the eyes, the nose, the ears, what they got treated with. But realistically, what we're likely to get at the first brush is whether they got treated or not, and whether it was BRD or not. And then if we want more than that, then I think we'll have to develop a more sophisticated recording system and actually try to target, in my opinion, animals that are coming through from breed associations where we can actually feed that data back to the breed associations. Because at the current time, none of that goes back into genetic evaluation. It's just kept for the records of that particular feedlot to know what's going on with their animals. So that break in the supply chain there, or the lack of feedback from the feedlot back up to the breeders, makes it a problem. So that's now being recorded. Is there a way that we can utilize this for genetic improvement? And so basically the guidelines recommended to try to integrate this information into genetic evaluations, and suggested a tiered approach to recording using the different levels that we found. So that enables flexibility for use in the data in genetic evaluation, and also doesn't require feedlots to do something dramatically different from what they're currently doing. And so I think what we would envision is that we would use both phenotypic and genotypic information in the genetic evaluation, and ideally animals that are genotyped, that are part of breed association evaluations, if we can get their data back from their feedlot performance or their offspring's feedlot performance into breed association evaluations, I think personally that's what's going to be needed to have accurate prediction equations for BRD, because I think we are going to have some type of a breed-specific component to make that work. So the tier 1 level of recording is basically what's already getting recorded: the animal ID, the lot information, in and out dates, sex, owner, origin, breed, that's going to be new, treatment information, and then that can be used to create a binary observation: yes, no, was treated, wasn't treated. And then the tier 2, and this is where the vets and the geneticists didn't have eye-to-eye feel as to what should be recorded. I think the geneticists wanted everything recorded, and the vets were a little bit more pragmatic in terms of, that's really, you're never going to get that. That's too much. You're asking too much. But if we could, we would like to get temperature. We'd like to get information on thoracic ultrasound and whisper evaluation and other contemporary group information. But at the moment, to be fair, this is kind of a work in progress. So Mark Enns is working with some of the breed associations to see if we can hook into their existing evaluation programs and try to get some of the phenotypic data back in terms of when these animals go on bull test, if we can actually also get disease information recorded at that time, so that we can start getting some phenotypes put together for the different breeds that can be used in evaluations in the future. One of the things, and this is a Larry slide, is that pen is likely to be a super important environmental factor in this case. If all of your neighbours are sick, or if your neighbours' wives have the flu, for example, then it's likely that you're also going to get the flu. And so you need to figure that in there, but there's a concern about breaking down the contemporary groups into basically ends of one, because they're not always going to be in the same pen. And so this is kind of a problem with genetic evaluation on traits like this, where you're getting phenotypes at feedlots where pens are not huge numbers. You can't keep the contemporary groups all together. So bottom line is, if there are large effect causative functional mutations, that would be the best of all worlds, and we don't know the answer to that yet. We haven't got the final results from the research team to know whether that's true or not. That would make incorporation of this into our genomic evaluations simple relative to the other approach, which is needing to develop prediction equations for all breeds and develop an ongoing phenotyping program. And so I'm going to let the data tell me what the answer to that question is, rather than just taking a punt as to what the answer might be. I think we do need to develop a standardized set of practical guidelines that could be used in industry herds and start getting those phenotypes into our breed association databases, if for nothing else, just to calibrate the tests that do come out. Even if they're large effect mutations, that you'd want to make sure that they're predictive in the breed that you're working in. You need to incorporate the susceptibility with an appropriate weighting into the breed economic indexes, and that's simply done when you're working with a large integrated operation where they are deriving all the value from the improved product quality and decreased disease incidence. It's maybe a little bit more difficult when you're maybe working with an index where the producer of the animal is not themselves getting a benefit from including selection against BRD in their selection index. And so somehow that has to be incentivized, or else a financially driven person would not include that in their index. And so clearly we can improve disease if we have the right selection criteria, even if it's a low heritability, but there has to be an incentive in there in order for that to get into the indexes. And does that look something like a backgrounding premium that we currently pay for producers that have appropriately backgrounded their animals to reduce the incidence of disease due to environmental conditions? Here we're doing a genetic improvement. How do we incentivize that? And that's really a little bit complicated by just the structure of the beef industry relative to, example, the dairy industry, where there's just that single index for the entire industry. So with that, I'm happy to take questions. I think I've got 5 minutes left. That was bad timing. I should have spoken for 5 more minutes, but I'm happy to take questions.
Actually, I'm surprised you didn't talk for 15 more. That's why I gave you 45 minutes to begin with. Any questions for Alison?
Right, so the GrowSafe, as you may be aware, has this thing called, I think it's called GrowSafe Beef, is that what it's called? Where it measures the animal and how often it's drinking water. And I think there has been some work done by GrowSafe where they've noticed that animals that are sick go off water a little bit ahead of obvious physical manifestations of bovine respiratory disease. So it's a potential early indicator. I guess our observation, because we did have GrowSafe data on the animals we were working with, is there's a lot of variability amongst animals as to when they drink and when they eat and how often they eat and how often they drink. And it's not always anything to do with whether they're sick or not. And I guess it was almost an overwhelming amount of variability. But I do know that that Alison and the GrowSafe team are looking at that, and I must admit that's really an area that we didn't have full access to the GrowSafe data, and so we haven't personally looked at all of that. We were trying to work on the genetic end, but if it's an early predictor, and you maybe pull an animal in because it hasn't drunk for a day, then it helps, then obviously that would be a good thing. But I don't know. We didn't see in our data an obvious trend, and we didn't really have access to all the data. We just had averages, which isn't that useful for genetic evaluation. I don't disagree. I guess that the reason I said it's of less value perhaps to the cow-calf person is that often BRD in beef cattle, as distinct from dairy, is getting seen as shipping fever once they've left ownership of the cow-calf producer and they've moved to the feedlot. So I absolutely agree there's industry good value associated with decreased susceptibility. It's a matter of how do you, because if you're going to select against disease resistance, you'll put less selection pressure on other traits that are economically relevant to cow-calf producers, and so somehow that needs to get incentivized.
So Alison, in the economic index work you did, I assume you estimated the genetic correlations between BRD susceptibility and all the other traits. And can you give us a feel for maybe the magnitude of some of those?
You'd have to you have to check with Justin. I'm not sure that I remember off the top of my head, but I don't recall that there was any obvious bad correlations that suggested that selecting for BRD or against BRD would have a bad effect on anything else. It's a little bit of a strange index because we were buying the calves at weaning, and so you'll see there's a negative economic effect or negative economic weighting there on weaning weight, which that's not a very normal thing, but it's because of the way that we structured that particular index is why that's a negative value. But I'd have to look at we did estimate the genetic parameters that I'd have to look to tell you for sure. Matt, thanks for that one.
Fair enough. I'm allowed one of those questions. All right, let's give Alison a round of applause.